thank you very much for the invitation to this wonderful workshop. I'm Miyer Patino Ruiz. I work in the University of Groningen, and my topic is providing synthetic cells with energy conservation modules that enable that synthetic cell to transfer and to achieve a metabolism. So the motivation for this work can be summarized in this question. So how can we help a synthetic cell to obtain nutrients from its environment? So how can we provide these strategies, efficient strategies of membrane transport in a synthetic cell? And that task, that the membrane transport protein task is actually important, and that importance is reflected by the high number of genes that in the minimal cell are dedicated to as membrane transport proteins, and these numbers actually comparable with other numbers in organisms with larger genomes. So thinking about strategies to provide with membrane transport in a synthetic cell, so we can think about passive strategies. So, for example, pores that are formed by toxins from some bacteria, they are continuously open and they allow the diffusion of metabolites or nutrients inside these cells. But we can also go a little bit more complex and have a bit more of control over the transport by using, for example, gated channels, which open upon a stimulation, particular stimulation, like presence of a ligand, a membrane potential of an osmotic stress. But if we want to construct synthetic cells that are autonomous, that are sustainable, that are able to keep gradients, we definitely need active transporters. That has, as Bert already mentioned, that has an energetic cost, and that energetic cost is in the form of ATP or in the form of electrochemical gradients of protons. And that particularly energy currency is what I'm going to talk about, how to provide synthetic cells with nutrients using the energy conserved as proton-motive force or as electrochemical gradients of protons. So when we think about proton-motive force, we know that that is established, for example, by primary transporters, by proton pumps, which use the energy from redox energy, or they can also use light or ATP to extrude protons out of the cell. And by that extrusion, there is the formation of these two potentials, an electrical potential and a proton chemical potential, or pH gradient, which is alkaline inside, and the membrane potential is normally negative inside. So the two components, these two potentials, form the proton-motive force, which is usually expressed in potential units. But there are other systems not based on primary transporters, but based on secondary transporters and enzymatic reactions, which can also serve to conserve energy in form of proton-motive force. And one of those strategies is present in these very nice bacteria and in some group of lactic acid bacteria, which are able to consume malic acid from the medium and exchange that compound for lactic acid. So it consumes a decarboxylic compound, a carboxylic acid, to form a monocarboxylic acid. And that's actually the basis of a common process in the production of wine, which is called malolactic fermentation. And what happens is that after the alcoholic fermentation, addition of these lactic acid bacteria will reduce the acidity of the wine. It will confer the wine a little bit more of buttery taste, buttery flavor. But what does it have to do with proton-motive force can be explained by seeing what is happening molecularly in these bacteria. So there, inside intracellularly, there is decarboxylase, an enzyme that produces lactate and carbon dioxide by consuming malate. During that decarboxylation reaction, protons are consumed, which results in the formation of a pH gradient alkaline inside. A second component, membrane transporter, secondary transporter, exchanges the lactate, the product of the decarboxylation, by external malate. And in that way, it keeps feeding the decarboxylase with the substrate and at the same time removing the product. And because of the difference in charge between the decarboxylic acid, between malate and lactate, then a membrane potential, an electrical potential, negative inside is formed. So together, these two potentials form our proton-motive force in two different steps, but in a coupled manner. So what we wanted to do, our goal, was to see if that system, if that simple two protein system was able or was sufficient to generate proton-motive force in a synthetic vesicle. And to do that, we reconstitute the malate lactate exchanger, and we encapsulate the decarboxylase in large unilamellar vesicles, which have a size between a diameter of between 100 and 400 nanometers. And what we wanted to test with those vesicles, with the synthetic lipid vesicles, was if upon addition of external malate and initiation of the malate decarboxylation pathway, then we could form a membrane potential and a pH gradient. And to test that, we use fluorescent probe to check, to test the membrane potential. This fluorescent probe diffuses in the membrane of the lipid vesicle, and then reports the formation of a membrane potential as a quenching, a fluorescence quenching event. And this quenching is actually proportional to the magnitude of the membrane potential, which allowed us to quantify the progress of the membrane potential in these vesicles. And what we observe is that the two proteins were absolutely necessary to produce a substantial amount of membrane potential and to keep it for longer times in comparison when we have absence of the decarboxylase. Then to test if we were able to generate, or if this system was able to generate a proton chemical potential or a pH gradient, we use another probe that Bert already mentioned, Pyronine, that to report the pH. And what we observe is that upon external addition of malate to these vesicles, we saw a prominent alkalinization. And together, they are telling us that it is possible to form proton-motive force. It is possible to conserve the energy that is released from the malate decarboxylation in the membrane of synthetic vesicles. But our goal was to actually provide nutrients to a synthetic cell, to a synthetic vesicle. So what we did was to couple, or was to incorporate another transporter, another secondary transporter that uses the electrochemical gradient of protons to internalize or to transport glutamate inside. This is called GLTP, is a proton glutamate symporter from Escherichia coli, and it's been actually studied by one of the members of our group. And what we wanted to test is if the proton-motive force from the malate decarboxylation system from these two proteins would drive the transport of glutamate inside the synthetic lipid vesicles. And what we observed is actually a very substantial transfer of glutamate inside at levels of accumulation of around 150-fold, meaning that with micromolar concentrations of glutamate outside initial concentrations, we can reach, after around 2 hours or for longer periods of time, we can reach levels of in the millimolar range inside, which are more compatible with the metabolism of a possible synthetic cell. But we want to, there are already strategies, experimental strategies to generate like electrochemical gradients of protons in lipid vesicles. And one of them is, for example, if you have a potassium gradient, potassium concentration gradient outward, and then you add varinomycin, which is an ionophore specific for potassium. Potassium will diffuse outside, and that will generate a membrane potential negative inside. If in addition to that you have an acetate gradient, an outward acetate gradient, the fraction of acetic acid that is in equilibrium with the acetate will diffuse outside, and outside it will dissociate into protons and acetate, and that will generate a pH gradient alkaline inside. So with these two diffusion potentials, we wanted to check if those those were sufficient to drive the transfer of glutamate, and to compare how we're those potentials in comparison to the Maillard decarboxylation system. And what we observe is indeed it is possible to generate glutamate transport driven by these two potentials, but the accumulation is much lower and actually is transient. So, and with this, we want to highlight that the Maillard decarboxylation system, together with proton substrate symporters, we can have a robust system, an efficient system to actually generate gradients in a synthetic cell, in synthetic lipid vesicles. And finally, we wanted to test also, like with another nutrient, a carbon source. In this case, we use a proton lactose symporter also from Escherichia coli. And we observe actually that driven by the proton motive force from the Maillard decarboxylation, it was possible to obtain also substantial levels of accumulation of lactose inside. We wanted to go a little bit farther. So what can we do with this internalized lactose? So we wanted to actually observe if it was possible to couple this proton motive force with a minimal metabolism. And to do that, we incorporated 3 enzymatic reactions that basically consume the internalized lactose to produce a reduction power in the form of this NADPH molecule, which actually the fluorescence of this molecule was served us to follow the progression of the whole pathway. And what we observed, it was actually that the proton motive force was indeed coupled to that minimal metabolism. And with this, I think we can, this is an initial platform, a starting platform for the incorporation of other modules or for the integration of other cellular functions into a more complex synthetic cell. So with that, I would like to summarize, perhaps saying that the modules of conservation of energy in the form of ATP that Bear already mentioned, in the form of proton motive force, are a starting step for the construction of synthetic cells that are able to keep an out of equilibrium metabolism, which is a hallmark of life. So synthetic cells that are able to work in a sustainable manner over time. And for that also active transport, we believe that is essential, not only to keep concentrations of nutrients in that viable concentrations for metabolism, but also to get rid of ametabolic products. We are working right now on then designing more like clever strategies. For example, not using proton amino acid symporters, but perhaps proton peptide symporters to then have free amino acids inside with the help of peptidases. And then what I showed today was enlarging lamellar vesicles, but then at which point we should consider perhaps have larger volumes or vesicles with larger volumes like giant lamellar vesicles. And that's going to be the topic of the next talk from my colleague. And finally, this is something that Bear already showed. We are working to couple the ATP and proton motive force conservation modules to have like nested system of vesicles. With that, I would like to thank all the people involved in this project, funding source, and of course you for your attention. And I'm happy to answer questions.
Thank you, Miyer. So I'm really impressed. My first thought, so my team and again, other folks in your Dutch Basink, I'm not sure, BASYC. I should know how to pronounce your acronym.
Basic, we pronounce it.
Yes, basic, of course. there is this ambition of building living synthetic cells. And one of the things that we imagine, so Kate Adamala and I are thinking about this, where we would take a mycoplasma vesicle, and we discussed, like Shige talking about the components of such a vesicle, put into that a transcription translation system and a genome, and hope that this just comes alive. But in order to make this work, you would have to generate the disequilibrium that exists in living cells between the inside of the cell and the outside of the cell. And that disequilibrium, as you and Bert have just shown, involves a lot of different components. Is this something, if you built such a cell just with cell-free extract, a membrane, and a genome, do you think that it would work on its own, or would you have to artificially make that disequilibrium in order to start this thing, in order to get it started?
Well, I believe that maybe doing it a bit of both. I mean, these systems, I mean, depending of the transporter that you use, you can generate certain disequilibrium on molecules, on some nutrients. Certainly with these systems, for example, with the Miyer decarboxylation system, you can generate already a disequilibrium of protons, which is already a source of energy. Then we can think about perhaps including other sodium electroneutral sodium proton antiporters, and then you have sodium multiphores, I mean, other electrochemical gradients of sodiums. But then if we're thinking about generating this balance of other nutrients, we have to start to incorporate other transporters, other components, and doing that in vitro. I mean, encapsulation or reconstitution of several transporters at the same time in the same vesicles, is rather complicated. But cells are actually like more efficient for that. So they actually manage to have a high density of proteins there in the membrane. So I believe it is possible. Yeah, it is possible using, like as you said, like a membrane of a minimal cell in a self-expression system. Yeah, I think generating a kick of this balance would be possible. Certainly, yeah.
How does your work fit in with the basic goal of building living synthetic cells?
Well, I believe, as I said at the beginning, yeah, maybe the complexity of the synthetic cell that we want to achieve is always a point of discussion. I mean, how autonomous should this synthetic cell be, right? Then either if you want to generate something really simple, perhaps you're okay if you just have like, let's say, passive ways of transport, and then you provide everything like amino acids and so on, but that would be a less autonomous cell. But if we want to generate something more autonomous and more closely resembling living cells, we need to start to incorporate more complicated things like active transporters, for example. Active transporter is I mean, those are proteins, relatively simple, but they're complicated in the sense that they need energy. So my contribution is maybe to providing this energy for the incorporation of these active transporters, such that we can generate a synthetic cell that is more autonomous, that is able to take the nutrients from outside, that is able to accumulate at these nutrients at concentrations that are actually compatible or with the metabolism, with a fluent and with a continuous metabolism in a synthetic cell. So I think that's the contribution that of these work in the synthetic cell. And of course, proton multiphores can have other. I mean, in cells, proton multiphores have other destinations or other purposes. it is believed that, for example, the machinery for cell division needs like this charge, this balance for proper location of some of the proteins. So I would say proton multiforce will be perhaps a way to help this cell division machinery. Yeah.
I have a question for you, Miyer. This is Kim again. Really nice work. I have a question about the lactate that's generated through your malate system. How does it get out? Does it get protonated and diffused like acetate, or does it need a special? I mean, otherwise it would build up and ultimately, I presume, stop your system from working. I don't know.
No, it actually well, it is exchanged by malate. The transporter works much faster when malate is exchanged by lactate. But yeah, if at some point it is high, I mean the accumulation of lactate is high. We also, I mean, lactate is biological membranes are highly permeable to this type of weak acids. So in principle, it could also go through the membrane. I mean, by passive diffusion, go out of the membrane. And actually, what happens in some type of lactic acid bacteria, which are which work really well under very acidic conditions, is that the transporter is actually able to work relatively fast in a uniporter mode. So malic acid maybe or malate can go through the transporter by like as a facilitator, and lactate, or in that case molecules as lactic acid, will diffuse via the membrane by passive diffusion.
Yeah. Okay. Thank you. And that, yeah, I'm the minimal cell. Of course, we generate tons of lactate, but I've never figured out how it gets out. But okay. Thank you.
Yeah.